This actually, particularly for germplasm, the viability and motility as well in case of sperms is very important, and it is important that we maintain the integrity of these physiological aspects of the cells so it can be used in a later time. So what are the tools of cryopreservation? I was talking to you guys a little about cryovials. These are typically how cryovials look. Depending on your need, they can be anywhere between 500 microliters to 5 ml or even larger. These are, they look like plastic, but these are high definition plastic, so they are resistant to a lot of cold temperature. They have a rubber gasket or sealing so they don't pop because of the internal or external pressure that is generated due to the low temperature. Next, we have this cryogenic cooler, and this is a very important tool when we are doing the gradual or slow freezing, even for vitrification. In our lab, we call this guy Mr. Frosty. So what Mr. Frosty does is we have a chemical compound, almost always acetone, basically an organic solvent that goes in between the white layer that you see here that has the space for the cryovials and the external layer. This Mr. Frosty is then put in minus 80 and negative 80 so that the solvent freezes. Once you are ready with your cells in the freezing media and you have transferred them in the cryovials, these cryovials are then transferred to cryogenic cooler, and this cooler is put in negative 80. So what happens is that the solution or the solvent in the cryovial gradually cools and attains a negative 80 degree Celsius, where it is frozen in 24 to 48 hours. After you have initially put this whole unit in negative 80, you transfer it to liquid nitrogen. This is a sample of how liquid nitrogen tanks will normally looks like. Now there are two types of liquid nitrogen tanks. One is a storage tank and another is a transport tank. Not all labs have universal hookup facility for the liquid nitrogen tanks. So in almost all cases, these labs will purchase liquid nitrogen from a commercial provider and then pour it into their storage tank. If you take a transverse section of this tank, at the lower panel shows what the tank looks like in transverse section. You have the main space where the pails, or we call the buckets, where your straws of cells or boxes of cells in the cryovials goes in. This is surrounded by a multi-layer insulation material, and that is sealed. It has a vacuum sealing, and the external is a metal shell. Next, we have a gooseneck. So this gooseneck basically is made of a styrofoam straw that gets into the storage unit through the cap. This basically prevents escape or evaporation of liquid nitrogen at a faster rate. There will be some, but it slows down the process. Then there is a cover plug and the capping that basically secures the liquid nitrogen in the tank. The storage tank is less complicated. The transport tank is less complicated. It has a narrow neck. It has definitely the insulation and the metal shell, but the space is arranged in a way that you can have liquid nitrogen with minimal evaporation, so you don't lose much during transport. Of course, when you are dealing with such cold temperature, you will need to protect your hands, so don't forget your cryo gloves that can resist the cold temperature, either it's negative 80 or liquid nitrogen. because if you're not careful, it can give you bad freezer burns. So you need to protect yourself too. So I'll talk to you briefly about cryopreservation that is used in vitro fertilization, and what are the unique techniques used to cryopreserve egg, sperms, and embryos. So why do we need to use cryopreservation? Why do we need to freeze egg, sperms, or embryos? This could have a varied application. It could be for the purpose of donor for couples who, for some reason, fail to produce their own eggs or sperms. It can be used to preserve egg, embryos, and sperm for potential future family. And I have seen many people coming to the oncology ward actually before chemotherapy will want to freeze their embryos or sperms or eggs because chemotherapy takes a toll on the fertility of that patient. So in this case, cryopreservation can be a unique tool when the patient recovers and is not able to have or have regained their fertility. They can use these frozen cells to start their family. It can be also used as part of family planning to delay having children at a convenient time. And sometimes under certain groups and ethical and religious groups, they avoid freezing embryos because the embryo is considered intact life. So they would go for freezing eggs and sperms instead of embryos, and to have their family at a later time point. So how is cryopreservation of eggs and embryos done? Cryopreservation of eggs is actually a three-step process where the individual or the person concerned will be given fertility medications to stimulate ovaries to produce multiple eggs. Once after the desired amount of time has been given following this stimulation, it's usually a minimally invasive 1-day minor surgical procedure where the patient comes into the outward patient ward and the eggs are retrieved in a surgical method. These eggs are then monitored in the lab for quality and to make sure that they have the desired morphological and physiological integrity, following which they go through the process of vitrification or being cooled in cryoprotectant, and then they're properly frozen using the necessary nutrients and self-raising media in liquid nitrogen that I already talked to you about before. Now, when the eggs are retrieved, they can either be stored the way it is, or they can be fertilized to form embryos and then preserved. In case of embryos, they undergo a comparatively more strict or stringent monitoring method in the lab, sometimes up to the stage of blastocysts, to make sure that cell division is happening properly, the cell structure looks great, the integrity is great, and once these parameters have been assured to be fine, that's when they take the embryos and freeze it for future use. In case of sperm, it is less stringent, but then because the motility and the count of sperm matters so much, semen samples need to use particular cryoprotective agents. But overall, what happens in sperm freezing is that the semen sample is collected either surgically or non-surgically, following which the spermatozoas are cooled to 5 degrees Celsius in presence of CPA or cryoprotective agents, and then they are frozen in liquid nitrogen to be used at a future time point. So what are the cryoprotective agents that are used? These are mostly low molecular weights that protect the sperms and decreases crystallization. You cannot use a high concentration of cryoprotectant due to resulting toxicity to the sperms. You need to have some internal that can penetrate through the plasma membrane, such as DMSO, glycerol, glycol, methanol, and and on top of it, they can also stabilize the cell plasma membrane proteins. Non-permeating cryoprotectants such as albumins, dextrins, egg yolk citrate, hydroxyethyl, polyvinyl pyrrolidone, and sucrose are also used to minimize intracellular crystallization and increase viscosity of the sap. Alrighty, with that, we can turn on our light bulbs and have some brainstorming session if you want.
So anyone who wants to ask any questions, maybe?
No questions?
If you have any doubt, you can ask.
Ma'am, I didn't understand properly the cryopreservation of egg.
Oh, okay. So what happens, because eggs are more secured physiologically, so it has to be retrieved surgically. But before retrieval of eggs, the ovaries needs to be stimulated with some medications so that multiple eggs are being released. This increases the chances of surgical retrieving of more number of eggs from the particular patient. So once the eggs are retrieved from the ovaries by a minimally invasive surgical technique, these eggs are screened in a lab to make sure they have the required integrity, morphology, and functional aspects associated with them. Once the screening is done, the best eggs are selected, which are then cooled in cryoprotectant. This step is called vitrification, where the cryoprotectant or the freezing media and the cells are rapidly frozen. Once it is frozen, this sample is then transferred into liquid nitrogen for long-term preservation and use in future. Does it make sense to you now?
Okay, ma'am. Thank you.
Could you follow Mrinal?
Yes.
Okay. Anybody else?
So a very interesting thing, like I was back in class 8 or 9 maybe when I first heard of cryopreservation. At that time, Madonna, she had, freezed her eggs.
Yes, that's
Yeah.
what I was. Yeah, Mitrajit, carry on.
Yes. So that was the first time, maybe like, almost like 15 years back when I heard first about cryopreservation. And then there was a gap. And then I heard that a number of companies like Google, Yahoo are asking all their female employees to go and do cryopreservation and, not leave their jobs at their prime stage. So it's very interesting. It's giving humans a lot of choices, actually.
Exactly. Exactly.
Yeah, I mean, technique-wise, scientifically, yes, you have this choice to, have the freedom rather
Yes.
to plan family according
Exactly.
to your convenience.
I would like to add that for the girls especially, this is a very good thing because now you don't have to worry about, you can just give some more time in your career and you can spend some more time. You can concentrate. You can like work for, and then you think for getting, going for family planning. So this is a very good method rather. So I think
Also, as you spend, like, particularly for, careeristic men and women, the quality of the germplasm goes down with age.
Right.
So if you
Yes.
want the best quality, you can freeze them at an
Right.
earlier age,
Yes.
and then use them
Yes.
at a convenient time when you have the financial and
Yes.
time-wise freedom to actually have a family.
So, like, I would like to interject here for one thing. Maybe a girl at her 22, she has cryopreserved her ovary, your eggs, and maybe at 45, she finally decides to have a baby or raise a baby because she is now financially very secure. So, my point is like, will a woman's body at 45 be able to have that child, or like, will there be any kind of, further thing,
Well, it depends
complications?
on how well she has taken care of herself.
Right.
I think the oldest female known to give childbirth was 67.
67. Yeah.
Yeah.
Right.
67.
Right.
So it depends, and sometimes even a person at 28-year old or a 30-year-old may not be in a physical state for childbirth. So
Right.
it depends from person to person.
Right.
But then
Actually,
we
yes.
also have this concept of surrogate mother in
Right.
case of people who
Excuse
cannot.
me, ma'am.
Yeah.
A person suffering from PCOD can have their egg preserved.
Yes, as long as the eggs are morphologically and cell-wise, they are intact, and they will go through screening, and that's why they retrieve multiple eggs.
So there was a
Is this possible?
there was one more
Yes, I
question
don't.
from a student. Like, the student wants to know, uh, how can he understand that eggs are well developed so that they can be preserved? So what are the methods, maybe if you know any, like, you want to share with us?
Yes, so they go through a screening technique, mostly under microscope. So
Right.
they will look for there are particular cellular integrity for a healthy egg. And then if the nucleus is not oddly shaped, and then some samples are used, maybe even fertilized, and see if they are allowing fertilization or not.
Right.
So there are a couple that becomes a long discussion in itself, how
Yes.
the screening technique is done.
So
But
anyway,
broadly
I
speaking,
think so.
yes.
His answer, his particular question is being answered.
Okay.
Sure, sure. So anyone else who want to ask questions, you can also ping me questions if you want. If you're not very comfortable, you can send me questions if you want. You can send Madam.
I think my email
Yes.
is
That
available.
will
Yeah,
be better.
I
Yeah.
think so. We can
That
finish
will
the
be
class
better.
here.
Yes, we can
Okay.
finish the class now.
Yes.
Okay. So thanks, Dola, for your. Thanks a lot. I think the students are much
There's one more
benefited by
question.
your
One more question. A
class
student
today.
has WhatsApped me. Like, we understand some students are not comfortable. It's okay.
Yeah.
So
Read the question,
there is a
Mithuji.
student who wants to know that how costly is the whole procedure.
Yeah. So it is costly, and depending on because a lot of it's not just the freezing, it's the retrieval, freezing properly, and there is some authentication of the clinic that is doing it. So at this point, I won't say it will cause you a arm and a leg or a kidney. It is not that costly, but yes, it is. I don't categorize it as a cheap method either.
All right. So I hope the question is answered. It's a cheap method. Anyone else who would like to question, or else we are closing the session? ठीक है, Dola di, thank you so much. Thank you so much
You
for
are
your time,
welcome.
precious time.
Okay, no worries.
Sure,
Take
sure.
care and
See
stay
you
safe.
again. Take care, Dola di.
Yeah.
Sure. Bye bye, everyone. Take care. I'm going to end the meeting now.
Yes, sir. Thank you.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you, sir.